So as you just heard, it is this session is going to be recorded. We have the previous recordings also uploaded on the RDKB Join the Conversation website. And this one will be there, I don't know, let's say in a couple of days after this is completed. So thank you, everybody. This is an RDKB hosted watershed webinar session, a science learning event for the Boundary Region. This is going to be the last one of this summer, hoping to start again in the fall. So check into the RDKB website and see what's coming up as we get closer to that September period. So you can, as I mentioned, check out our past webinars at the RDKB Watershed Webinar, Join the Conversation link. I believe this is going to be our 7th one. I know that we had some in-person ones and it's moved to the webinar series as we've gone through the COVID process. So yeah, it's been a pretty neat way of being able to hang on to so much of this information from this region. So as we begin, I acknowledge the First Nations and Indigenous influence in the Boundary Region and the First Nations traditional history here in the Boundary Watershed. Yesterday being National Indigenous Day, I see this as a time for reflection and strength. I know I've learned so much. I know I have a lot to learn about the traditions in this region, and I am so grateful for those that have been sharing their knowledge. I respectfully appreciate that the land on which many of us gather and is referenced in this webinar is a converging traditional and unceded territory of the Syilx, Sinixt, and Secwepemc peoples. We're grateful to have this collective opportunity to share our knowledge and information via this webinar, both from our presenters and our attendees. And that's sort of a big part that I really wanted to put in. That transfer of knowledge on both sides is really important here. And there's many of you that have signed in that have some very deep knowledge of the Boundary Region. And we have this opportunity to be able to share that knowledge and those experiences and perspectives with Stephanie and Jolene, our presenters. So I really encourage, whether it's a chat box or questions, to give us some information about your area or what are the questions, what are you seeing out there? So you never know what bit of information, but it might be that one thing that prompts somebody else's action. So I encourage it. And a lot of what we've been looking at over the last year or so has been sort of that climate change riparian actions, that land use impact. So I really see this aquatic invertebrates component as a great fit for that. So my first experience with Cabin, the monitoring system, was in Tumbler Ridge. I had the opportunity to join in with a crew on their annual survey, and it was fascinating. One of those experiences that's really struck with me. I was able to see an incredible array of aquatic invertebrates in the control system, so that's your untouched watershed, and compare it to what was found in the more impacted streams, and being able to use what we found to tell us a story. And also it was pretty neat to see some that the story was the same as what the control stream was. So that was also equally as exciting. I didn't know much about the diversity and indicator capabilities of aquatic invertebrates. So ever since then, I just wanted to keep passing on this information. And so when I read about the 2017 Okanagan-based document reference, this is a big title, reference model supporting document for cabin analytical tools, it had a contact person's name, and it was Stephanie Strachan. And so I cold emailed her to say, hey, we're looking to try to do these webinars, and would you be interested? And Stephanie, lucky Jolene, got me in touch with Jolene as well. And so here they are. So thank you very much, both of you, for participating and for being willing to take this on. So for those of you that have attended my these webinars before, you're going to recognize this poll. And so one of the things that we do like to find out is a little bit about, where are people signing in from? Where have we got people? Where do you live? Do you live or do you work in the area? Are you familiar with the boundary, or have you been here? Is this where you're based? So there we go. We've got a couple of people signing in there. So it's great for both myself and Stephanie to be able to understand, what's some of the background. So you'll see a couple of polls as we go through this presentation, just trying to better understand, who do we have in the audience. So right now we have 12 people. So I guess 9 people signed in, and we've got, here we go. I'll just end this poll right here. Ew, 50-50 for living and working, for living in the boundary. And so, so this is sort of that equal point almost, yeah, it's 50% on both sides. So I'm Christina Anderson, watershed planner with the RDKB, and I've been here for about 2 years, and I've been really enjoying getting to explore the area and learning so much about the boundary region. And this is a big part of wanting to sort of help move some of this information through to out to so many of the community members within the boundary of so much of the research and work that's been happening in our area. So I'll introduce our presenters. Stephanie Strachan works as an environmental monitoring science, biological assessments with Environment and Climate Change Canada, freshwater quality monitoring program. So that's the federal Canada. Stephanie is a BSc in the Great Lakes Co-op study from the University of Windsor. And interest in sediment quality guidelines. I know that's been a big part of a lot of conversations in this region. MSc in zoology from Western Ontario, focused on leaf pack invertebrate communities in the Fraser River, which was where the cabin program was born, and has been an environmental monitoring science with the ECCC for 25 years and currently lives in Vancouver. So Stephanie, have you spent much time here in the boundary region? Are you familiar with this area?
Just unmute myself. Yes, actually. Luckily, the one benefit of this job is I've been all over the province. I have been to the boundary region. Love it out there. We have, and you guys will see soon, Jolene will show you, we've sampled some sites around Granby and in the Kettle, and we have some long-term monitoring sites in the Kettle. So yes, I have been there.
Thanks very much. Jolene Raggett works out of the Nelson office, so pink building for any of you Nelson knowledgeable folks, as the environmental impact assessment biologist with the Ministry of Environment Climate Change Strategy, so the provincial for the province of BC. Jolene has a BSc in biology from UBC and a post-baccalaureate diploma in environmental science from Kaplan University. Jolene has been working in the Kootenays with Emily since 2003 and with the cabin program for the past 5 years. So Jolene, is your part of your area of the boundary region, and has that been when would that have started being the case?
Well, when I worked as an impact assessment biologist, I was focused mostly just in the Kootenays. Okay. So I actually coordinate the provincial biomonitoring program now. So most of my work group is in Victoria, but so I work all over the province, not just in the Kootenays. So I have also done some sampling in the boundary.
Oh, super! Thank you. And it's been pretty neat, sort of seeing some of the different sites that have been sampled in this region. So I'm just going to really quickly not spend a lot of time on this, because I'm going to say that most of you are very familiar with the Zoom platform. So for the chat box, and we'll see, we've got one chat in there. So yeah, for the chat box, if you've got any technical issues that you want to be able to alert me for, I'll be monitoring that chat box throughout the whole presentation. And also, if you've got any comments, maybe there's something that you agree with, or there's a component of what they're saying that you can see in this area. So some of that sort of information in the chat box. The Q&A, we're asking that all questions that you have about the presentation are focused in that Q&A box. You can see over here at the right-hand side. And if I see a question that is going to be able to really help understand the presentation, I will interrupt Stephanie or Jolene. Otherwise, we'll leave the Q&A to the very end of the presentation, and then they can they can respond to them. And then in the event that you want to say it verbally, and we may open it up depending on how much time we have, to raise your hand, and then I can unmute you on your side. So you are going to be muted for the presentation, but there's no issue at the end of it all for us to be able to unmute you. Okay. I will pass it over to Jolene to set up the presentation.
All right.
Thank you very much. And yeah.
All right. Are you able to see it?
There are.
Okay, great.
Good. All right. So Jolene's going to operate the presentation. We're doing this jointly. So Jolene and I work very closely together, and we usually do this kind of a presentation independently. This is the first time we've actually done it together. So you guys are are going to be with us while we figure out how to do that together. So I worked with, as Christina said, and thanks for the invitation to do this, but I work with Environment and Climate Change Canada in the Water Quality Monitoring and Surveillance Group. So my focus, as Christina indicated, is biological monitoring. So I am actually the cabin regional lead for Pacific Arctic Athabasca watershed, which is huge. I'm going to give you a little bit of context about cabin and then about a focus in the Kootenay. But I first want to acknowledge that I'm sharing this presentation as a settler living in the unceded territories of the Squamish and Musqueam peoples in Port Moody. So I'm not in the boundary region. I didn't answer that poll, so I would have taken you over the top to outside of the boundary region. I would have been your 52% or whatever it would have been outside of boundary. But I wanted to be able, I take advantage of the polls that Christina's got here. And because cabin's in the title, if people aren't familiar with cabin, they're probably going, what is this? And then also, if you're not familiar with benthic invertebrates, you might be going, what is this? So Christina's gonna throw up some polls at a couple of places in the presentation just to help Jolene and I with a bit of context about what people in the audience already know. So then we can maybe tailor our message a little bit as we go. So maybe before we dive too deep, Christina, if you want to put up another couple of polls just to give us, for me personally, I'm curious how many people even know what cabin is or if this title seems bizarre to you. How familiar are you with cabin biomonitoring program? Do you know what cabin is? You spent many years doing it. Are you familiar with it? You've used it before, or you don't know what cabin is? I'll let you control. You can probably see.
Yeah, so we've got 10. We're almost done for the voting. And I'm always impressed with the number of people that are signing in and with being very new to some of these systems. So I think we just got one more person that looking in to vote, and I will share. That's right. So of course I
heard of it. Okay, awesome.
Seen it.
So this is super important for me. So I'm glad I know this right off the bat because I will be giving you a lot of introductory information about what cabin is. So this is awesome. A couple people are familiar with it, so that's good too. So excellent. I think that I hopefully will give you some good information about that. You can go to the next slide, Jolene. Oh, it's probably the poll questions. You can go to the one after that.
Oh, do you want one more poll?
We can do that. Do you have that now, or we can do it after?
Either one.
Okay. Okay, so familiarity with fresh water. So everybody on the call can give us a bit of an indication about if you are connected with the freshwater, either in the work that you do, and if what you know about types of aquatic invertebrates in your region. So also help us with a bit of context when we're going through our presentation.
Yeah, and thank you very much, everybody, for this. And I think it also helps the attendees to know who also is in the audience. Yeah. Just another 10 seconds here waiting. Oh, we're close there. And here I will end the poll. And there we go. So definitely majority of people either work or spend time around that freshwater systems. And yeah, so somewhat knowledgeable about the aquatic invertebrates, and to know, I don't know.
All right, well, that's good because we'll spend some time on that too. Excellent. All right, let's go to why are we doing biological monitoring? Most everybody's probably familiar with the concept of water quality monitoring. Most people sounded like they did work around freshwater, so they're probably familiar with that. When we do water quality monitoring, usually we're just taking water samples, we send them to a lab, they'll analyze them for a variety of parameters, and then we'll compare them to guidelines like nutrient guidelines or metal guidelines, and we'll say whether or not that, that sample is good, means it doesn't exceed any guidelines, or if it's bad because it's exceeded some metal guidelines or nitrogen guidelines or something like that. So that's usually what everybody's familiar with. And since 1990, 85 actually, so it's been quite a long time. The federal government, the provincial government have had an agreement to jointly manage a long-term water quality monitoring network. Oh, thanks, Jolene. Which means we together decide where we want to do long-term monitoring, what are the issues at those particular sites. We send samples to the lab, sometimes bi-weekly, sometimes on a monthly basis, and we compare them to guidelines or objectives that protect freshwater health. So the assumption is that, you know, and people do this all over the world. They do this in countries all over the world. And the assumption is that if they don't exceed guidelines, then the system must be healthy. But we know that there are lots of things that could be affecting ecosystem health that cannot be measured by water quality samples. So like, even though it's used a lot, there are countries, including Canada and the US, that are looking at biological organisms like algae or macrophytes or zooplankton or fish or bugs to directly measure biological health in the ecosystem. So biomonitoring usually complements water quality monitoring programs, or it can be used to verify the kinds of problems you might be seeing with water quality monitoring, or it can be used to identify undetected problems. So the things that you can't actually measure with a water quality sample. Sometimes the biological indicators are used as early warning indicators. So maybe we can see something happening in the community before a guideline exceedance happens. So we've recently incorporated biological monitoring into our Canada-BC water quality agreement. So Jolene and I work very closely together. You can go to the next slide, Jolene. So one of the reasons we're incorporating biological monitoring is because we want to look at cumulative effects. So in other words, what can we see from the combination of things that happen in the past and currently related to human activities, as well as natural processes? So we know chemicals can interact with each other, and the effects caused by that can be different than if they were in the system by themselves. We know that water samples can only measure contaminants in the water at the time of sampling, but what if a spill happened last week and we're sampling today? Are we going to pick up that problem? Will we always know what contaminants we should be measuring at a given site? We know habitat degradation and invasive species will affect the ecosystem. And these things can't be measured with water quality samples. And then of course, there's the changes to water quantity or flow. And we've seen that from climate change. And we know that the ecosystem is greatly affected by these processes. So this is one of the reasons why we've incorporated biological monitoring into our long-term water quality monitoring agreement. And we'll talk about the 2 kettle sites in this presentation, which Jolene will get to. So I think now we can ask another poll before I get into the bug questions or the bug presentation. So I'll let you throw that up there, Christina. What are some of your favorite aquatic invertebrates? Now, not everybody was familiar with aquatic invertebrates, so here's some examples. Beetles, snails, mayflies, worms, caddisflies, mussels and clams, dragonflies, leeches. So this is what we're talking about when we're talking about benthic aquatic invertebrates. So see if any of these are your favorites. If you have any favorites, I can't wait to see who checks leeches.
Now I had to separate it. So, like, look, worms and leeches are in the same group. Put them together. I'm like, I cannot put worms and leeches in the same group under favorite. So yeah, that was totally 0 of 7 for leeches so far. 0 of 8 for leeches so far. Leeches is not being pulled on as a favorite. Oh, we're so close. Oh, we got it 100% voted here. End polling.
Ooh, nice.
Oh, leeches. Somebody got leeches.
Somebody picked it just for fun. They didn't want them left out. I expected the dragonflies to be among the top ones, so that's awesome. Beetles, wow, cool. And worms. I'm thinking there might be some fishermen in there with some mayflies and caddisflies at the top. Awesome. Oh, stoneflies is down at the bottom. I missed it. I saw the number, but I didn't see the bar.
Sorry, I can put that up there again.
Can you? I can only see 33%. Okay, that's good. Lots of stoneflies too. They're evenly distributed with snails and beetles. Awesome.
Okay.
Do you have another one? The biomonitoring knowledge, is that one here too, Christine?
Do.
Okay.
And
Okay, so I feel like I'm gonna know the answer to this just based on the other questions we've asked already, but if you guys could answer this one too, what is your level of knowledge about aquatic biomonitoring? You have a freshwater background, so you're pretty familiar with it. You've heard about it bits and pieces through the course of your work, or it's very limited and you're gonna learn a lot about it today.
And it's kind of neat, so we've got a poll at the very end here as just to sort of try to look at maybe some of the things that people have learned. And we're actually going to be doing a survey at the very end of this presentation as well, to be able to look at that as well. Okay, we're almost there. And I'll close it now.
Okay, good, awesome. So I think that Jolene and I hopefully have tailored the presentation for the people that have moderate and limited knowledge about biomonitoring. So this is good.
Yeah, and ask questions as we go through.
Great. Okay, so because of the limited knowledge, I thought that I would spend a bit of time talking about bugs. So most of what we're talking about here are benthic aquatic macroinvertebrates. So these are the bugs that live at the bottom of the streams and rivers. And most of these bugs are in their larval stages, which means they're in their immature stages, meaning they will eventually come to the surface, emerge as adults with wings, and then disperse. So I wanted to kind of give you some associations on this slide between the larval stage, which is what we're looking at for biomonitoring, and what they eventually look like as adults, which is probably what most people are more familiar with, and then a few little facts. So in the top left with the dark blue circles, those are mayflies. Mayflies are among the pollution sensitive bugs. These guys have 3 tails usually. So if you're and they're small. So if you're looking at them, you usually will see 3 tails. And these guys also have pretty gills along their abdomen. So can you see you probably can't see my screen. Can you see my cursor? No, you can't, but that's okay. This kind of longer end part of their body is their abdomen, and that's where their gills are. And then they kind of flutter in the water, and that's how they pick up oxygen. When these guys emerge, they only live for 24 hours. They lay their eggs and then they die. I came from Ontario, and I haven't seen this here, but in the Great Lakes, when these things emerge, it's massive, and you can shovel these things off of your driveway when they die. So what you see here in this car is a massive mayfly emergence that usually happens in June, despite the fact that they're called mayflies. And yeah, everybody in the Great Lakes area thinks they're really gross, but they're actually nice. They're good indicators of ecosystem health. So as much as everybody hates them, they're actually a good thing. And this is what they look like as adults. So a lot of fly fishermen might be familiar with these guys here. Down in the bottom, the light blue circles, these are stoneflies. These guys are much bigger. These guys have 2 tails as larvae. They have gills in their armpits. So they don't flutter in the stream. They actually do push-ups to try and move water past their gills. And these guys are larger, as you can see the adult on the person's thumb there at the bottom. And again, a lot of fly fishermen will tie flies to look like stoneflies because this is what the fish are eating. If you look at the top right, the orange circles, these are caddisflies, another pollution sensitive organism. These guys are really neat because they make their own houses. So they make their houses out of sand grains or sticks or pieces of leaves or whatever. And you can see all of those little cases on the rock in that top picture. Those are caddisflies. And so they stick their little heads out of their houses and they kind of catch food as it floats by in the stream. Now, what's also interesting about these guys is people will use their skills of making houses to make jewelry. So they will put this larva in little containers with precious gems and gold flecks and stuff like that. And they will make the houses out of these precious stones that you can see in the picture here. And then you take the little organism out, you take the case and you can add it to your jewelry or your necklace or earrings or whatever. So, and these guys kind of look like moths when they're flying around. Again, one of the pollution sensitive indicator organisms. In the middle, these are the dragonflies. Now you don't often see them in the streams, you see them more in wetlands, but these guys are big and these guys are predaceous. So you can see, hopefully you can see it well enough that in the middle, the bigger picture, that guy's eating a fish. So they have these giant mouth parts that protrude and then grab their prey and then bring it in. And if you're familiar with the movie Alien, the alien creature was modeled off of this dragonfly. So it had this protruding mouth part that would grab things and then eat it. But that's, so everybody's favorite was a dragonfly. It's pretty scary as a larva. And then the bottom one are the dipterans. These guys are our pollution tolerant organisms. These are things like biting midges, which is the bloodworm you see at the bottom. And beside it is what it looks like as an adult. They're the black flies. So what you see, all those little critters on that rock there, those are black fly larvae. I actually really like black fly larvae, not the adults, just the larvae. They stick their bums on this rock and then their whole body sticks up on the rock, and they like the really fast flows because they have these fans that collect food and that stick up in the flow as it's flying over. They actually, when you look at them under the microscope, it looks like they have Mickey Mouse ears, but they're actually fans. And that other creepy thing in the bottom corner, which looks like a worm, is actually a cranefly larva, Tepulidae. So these are our pollution tolerant type organisms here. And it's very general in terms of how I've described them, pollution sensitive, pollution tolerant, but it's generally how people talk about the water quality indicators. So that's a little bit about the bugs. But the bugs are used worldwide. Like, they're super common for people to use as water quality indicators. And it's the focus of the cabin program, which I still haven't explained, but I'm getting there. So they're popular for a lot of reasons. First, compared to fish, they're relatively sedentary. So they're living at this site. So we know that anything that they've experienced over the course of their life is reflective of that site, unlike fish that can move quite a lot in a stream. Compared to algae, they're relatively long-lived. So most of them will live in their larval stages between 1 and 3 years and then emerge as an adult. So anything that's happened over the course of their lifetime will be reflected in that community. And because of, you've already seen the diversity of benthic organisms, some of them pollution tolerant, some of them sensitive. They respond to all kinds of different stressors in all kinds of different ways. So we can use them as indicators for just about anything once we kind of know how they respond to it. And unlike fish, you can find them everywhere. They don't respond to fish barriers because, like I said, they grow wings and emerge and then go lay their eggs. So you will find them in any system. They're a key part of the food web. As you've already seen, they can feed on fish, but most of them are food for fish. And then others will be eating algae off of the rocks or off of the leaf litter and other woody debris. And because they're used all over the world, the research is well established, the protocols are well established, but that's also part of the challenge, is that the protocols, because they're used everywhere, they're used in different ways. And so the data is not always comparable. You can go to the next slide, Jolene. So this is how CABIN came to be. So CABIN is the Canadian Aquatic Biomonitor Network. It was established by Environment Canada at the time. We've changed our name since, but it was Environment Canada at the time because we were interested in pulling together biomonitoring information at a large scale. And when there was a patchwork of stuff, there's a little bit here and a little bit here and a little bit here. And we noticed that we couldn't actually pull that data together because it wasn't collected in the same way. So it wasn't comparable. So in the mid-1990s, we established standardized protocols that could be used for almost anywhere for the assessment of aquatic ecosystem health. So it started as a research project in the Great Lakes, and then we tested it out in the Fraser Basin in streams and rivers. And it was done initially to complement much of our water quality and sediment quality work that was being done. But once these standardized protocols were established, we were able to share data with anybody else doing the similar kind of thing and then collaborate in areas of joint interest. And this is how CABIN was born. So CABIN's based on a network of networks approach. So we've got online tools, a shared database, a training and certification program to ensure that the data is comparable and consistent. And it's managed and maintained by a national team of Environment Canada staff. We focus on key components like the training program or the database or the taxonomy standards or the QA quality assurance standards, that sort of thing. But we also work with other partners in the network to further develop aspects of cabin. So we have other research and development things, like we've got a wetland sampling protocol, but we're currently working on wetland assessment metrics. We've got a Great Lakes deep water lake protocol, but we're working on a protocol for small lakes. We're working on a protocol for large rivers. We're working on standards for the inclusion of GIS-derived landscape information. We're working on a northern biomonitoring program, like how can we actually get more information in the north where it's so difficult to get to these places. And also we're looking at the potential inclusion of DNA-based information in the cabin program. So there's a lot of stuff going on that's not fully adopted into the program yet, but we work with our partners on helping to develop this. And once we get to a point where we might be ready to implement it, we have a science advisory group with internal scientists and external experts in the field that kind of review these different steps just to ensure that we maintain that scientific rigor and credibility. So that's the cabin program in a nutshell. Now I'm going to dive into the parts a little bit. So most of the work that's being done is in streams and rivers. So the protocol that we have established collects a lot of different pieces of information. We collect a lot of information for just about the site, like lats and longs, stream order, elevations, things like this, a description of what the site is like, the habitats present at the site. We have a standardized process for how we collect the benthic invertebrate community. We do channel measurements like depths and widths and velocities, things like that. We also looked at substrate, like how big is the substrate? Is it buried in a lot of fine sediment? Like these are the kinds of things that drive what kind of bugs you will find at your site. And then we've also started to look at landscape information like bedrock geology, climate, topography, because that stuff is also a driver of the community that you're going to see. Cabin's usually, as I mentioned, part of a water quality monitoring program, but if it's not, and people are just interested in looking at their backyard bugs, then we also have some suggestions for some basic water quality parameters that you should probably collect at the same time. And all of this is to ensure that, you know, the data that Jolene collects is consistent and comparable to my data, which is consistent and comparable to maybe Christina's going to go out and do some sampling, and then we can actually all compare our information. Next slide, Jolene. So the one thing that we noticed that was, as we started this program kind of in the early 2000s, is that training and certification was going to be really critical in order to ensure data comparability. We also realized that with these online tools, people were needing guidance for how to use the database, how to use the analytical tools, and what does it mean. So we developed an online training program, which has online modules, but complements a 2-day hands-on field certification component. And we're trying to always improve this because in the beginning, our network was mostly government people. So it's people in my office, people with DFO, people in Jolene's office, and industry. But then we found that more and more community-based monitoring groups and academia and First Nation groups were wanting to participate in this as well. So we're trying to modify our training delivery so it is accessible to everybody who's interested. Next slide, Jolene. So Jolene's going to talk to you about one of the big benefits about the network of network approach in a minute, but I'm just going to talk more generally. The benefit of the network of networks is that it allowed the work that I'm doing to be combined with the work that Jolene's doing, to be combined with anybody else doing work, primarily the collection of reference data. So reference data from sites that are minimally impacted by human disturbance, because that's the data we use to build these assessment tools. And if any one of us tried to do it by ourselves, it would have been really quite expensive. So there is a huge benefit to collaboration. So this pie chart, what this shows you, this is based on data from September of 2019. So it's a little bit outdated, but this just shows you the current, most current I have, distribution of by sector of the people using the cabin tools. And it's probably not surprising to you that 23%, the largest chunk, is the federal government. The other large chunk is actually industry. So they're using them for environmental effects monitoring and impact assessment. But the other pieces are growing, particularly the NGOs and academia and indigenous groups. Those are growing. And the governments are becoming a less prominent part of this network. So maybe if we go into the next one, this is going to take a little bit of explanation. So the benefit of the network I mentioned is this use of an assessment model. So CABIN is based on what we call a reference condition approach. So we use this reference data that we've all collected to develop these bioassessment tools. And we develop these tools at a basin level. So Jolene is going to talk to you about the Okanagan, Kettle, and Similkameen Basin. That's the level we're talking about. And what we're trying to do with this information is, with all of the data that's collected, we're trying to understand what's the relationship between the bugs and the environment in these clean sites. And once we know that, that's essentially our predictive model. So if you look at these two groups of bugs on the slide, you don't need to know what the images are. You don't, they are particular images, but just looks at the shapes and the colors of them. And you can see that those two clusters are different. Those are two different community types. And so essentially what we're trying to do is we will look at the first one on the left and we'll go, oh, those tend to be found in headwater streams, tend to have low conductivity, quite shallow, low flow. And then we look at a different kind of community and say, oh, those tend to be found in mid-sized streams with high conductivity, and they tend to be deeper and faster flows. That's the simplistic description of a predictive model. So if we wanted to go to a site to do an assessment and know, okay, is this good or bad? We would look at those environmental features, we'd plug them into the model, and the model would say, your site should look like this. And then we make that comparison. So that graph you see on the far right, that's an ordination plot. That's a way of us to throw all of that aquatic information into a graph and show it in 2 dimensions, which you can imagine is statistically challenging. But just imagine that every single white dot represents one of those community clusters. So all those white dots are all reference sites. You can see they're all in a big cloud. Your site, if it was not impacted by any kind of human disturbance, should be near the cluster of white dots. But we've drawn confidence ellipses around it so we can kind of define the degree of stress or the degree of impact. So if that blue star in that graph was, let's say it's the Kettle River site, and we plotted it with our predicted reference sites. So this is what it should look like if there's no stress on this environment. It should be close to it, and it's not. So in this particular instance, it's in the orange zone. In the orange zones, we're saying that's divergent. It is outside the 99% confidence ellipse. So that's kind of different, and we want to figure out why it's different. If it was in the yellow zone, it would be mildly divergent. It's a little bit different, but we won't be too concerned yet. We'll just keep an eye on it. If it was in the green, it's good. It's just like the reference. It's exactly what we expect. If it's in the red, clearly something is wrong and we need to figure this out. Let's go to, maybe just before you switch, Jolene, I did want to mention that after we do this graph that's on the right, the tools in CABIN allow you to dive deeper, and they allow you to say, the probability of seeing worms is this, the probability of seeing mayflies is this, and you can dive deep into what you expected to see and compare it against what you actually saw, and you're like, oh, I thought that I was going to see all of these pollution sensitive taxa and I didn't. Is there a contamination problem here? Or look at all of these burrowing organisms that I saw. I didn't expect to see that. And you're like, maybe that's an indication you have a sedimentation problem. So you do need to do a little bit of investigation about freshwater ecology. So CABIN can't spit that out for you, but it gives you the tools to dive a little deeper. So now you can go to the next slide, Jolene. So it will also, it'll look at diversity. It'll calculate how many different kinds of bugs you saw. It'll tell you what percentage was pollution tolerant, what percentage was pollution sensitive. It will give you a quantitative measure of how similar your site was to what you expected it to be. So CABIN will help you to drill down a little bit into the potential causes, but you still need to do a little bit of background research in terms of what those different bugs mean. So that's the overview of what CABIN does. And now I'm going to hand it off to Jolene, because she's going to actually give you like a real example of how this operates in the boundary region.
All right, great. Thanks, Stephanie. That was interesting even for me. Just because you presented it in a very clear way. So now that you all have a bit of background information on the cabin program, as Stephanie mentioned, I'm going to share some information that's more specific to the boundary region. So you can see how cabin tools can actually be used as part of a watershed assessment. So this map here is taken from our provincial biomonitoring webpage. It shows the boundaries for all the reference condition approach models that Stephanie mentioned that are currently available in BC. So the models are maintained, they're updated about every 5 to 8 years. So that means we bring in data from new reference sites that we've sampled during that time, or reference sites that we go back and we collect repeat samples, and we build those into the model as we update it. So the Okanagan model is blue 1 shown in the bottom. So the Okanagan model was most recently updated in 2018, and it was updated, it used data from 69 different reference sites. So the reference sites are shown on the map here. They're distributed across the entire Okanagan. So there's samples collected in the Similkameen, the Okanagan, and in the Kettle watersheds. This map also shows the different types of habitats that were identified across the Okanagan. So we used GIS techniques to try and identify the different types of habitat. And then we selected reference sites within each type. And that makes sure we have good representation of the range of benthic communities across the Okanagan. When the model was built, there were 4 different types of benthic communities that were identified. So those are the groups that Stephanie was talking about. And so those 4 groups were correlated with predictor variables. So those are the habitat characteristics at each site that are used to predict what type of community we'd expect in a healthy watershed. For the Okanagan model, these included variables. There was like channel characteristics, that was depth and substrate size, bedrock geology, elevation, and also local climate conditions. So here's another map that shows the Okanagan model in blue. So that's again from our provincial biomonitoring webpage. This shows all the reference and test sites sampled by Environment Canada and the BC Ministry of Environment within the Okanagan. So this map doesn't show sites sampled by others. So we don't have sites sampled by First Nations or industry or stewardship groups, but those actually can be found on Environment Canada's website. They have an activities map that I'll show a link later, and you can connect in if you want to see what other sites are sampled in that area. So I'm going to move into sharing some data from the Kettle River, which is shown in the map. So I think probably most people are familiar with the Kettle River, but it originates in the Monashee Mountains. It flows south past Midway and then Grand Forks before it enters the Columbia River in Washington. There's 2 sites on the Kettle River that were established as part of the Federal Provincial Water Quality Monitoring Agreement that Stephanie mentioned earlier. So we've got Ket 01, and that's located at Midway. The second site is Ket 02, and that's located right at Grand Forks. So cabin sampling began at these sites in 2007, and the cabin samples are collected every 3 years. So we've got data between 2007 and 2019. So the sites were included in the federal provincial agreement because the Kettle River is transboundary. So it's really important that we're tracking water quality that's entering and leaving BC. At the time they were established, there were big concerns about the effects of mining in the US, and also about water shortage issues, and then concerns about run-of-river dams within the watershed. So in addition to those issues that were listed in the agreement, there's other land-based activities in the Kettle watershed that have the potential to affect the Kettle River, so forestry, agriculture, urban and rural development. So it's important to keep track of changes in water quality and aquatic health. So I did an analysis of the cabin data from the 2 sites using the Okanagan cabin model to share as part of this presentation, and just to demonstrate how cabin can be used as part of a watershed assessment. So here is the first site. So Ket 01 is located at Midway. It's just upstream of the Florence Street Bridge for people that are familiar with Midway. And the image here shows the view from the site looking upstream. So this is what your outputs will look like when you analyze sites. So we have data from this site from multiple years. So we've got 5 sets of data. Based on the habitat predictor variables at this site, the Okanagan model compared it with benthic communities found at similar sites in group 2. So the group 2 sites contained the most similar type of habitat to this site. The table in the upper left corner shows the overall site assessment for the site each year it was sampled. So you can see the green and the yellow here. So for the first 2 years, 2007 and 2010, the site was mildly divergent. So it's a little bit different from reference, but it's not overly concerning. It's fairly close. The benthic community is fairly similar. We see a bit of a shift in 2013 where the community moves and it's characterized as being divergent. So 2013, 2016, and 2019, we see the community is different from the reference site. I've shown an example of the ordination plot for the most recent data. So this is 2019. So this shows you what an ordination plot that you, when you run a site in Cabin, what it actually looks like. So we see the reference sites here in the middle in green, and then the test site is out in the yellow ellipse. So when we see a result, results like these, we see that there's something that's different about the benthic community, but we don't know why it's different. So you can do further investigation to explore what aspects of the community are actually making it different from the reference sites. And I'll get into this on the next slide. Okay, so there are web-based tools in Cabin that will let you run the site and look at specific metrics of the benthic community that may give you a hint about what's happening and why the site is different. So for example, if we have a lot of nutrients present in a stream, we might expect a large increase in abundance. So there might be a lot more organisms present because there's a lot of nutrients there. If we've got high levels of contaminants in the water, we may expect to see a decrease in sensitive taxa. So the EPTs, I don't know who's heard of them, but like the Ephemeroptera, Plecoptera, Trichoptera. So those are the sensitive taxa. So I've included a table here just to show you what some of the metrics are. There's a lot more you can run. Just as an example of the outputs you can get through Cabin. I haven't done a thorough assessment of the data. I just wanted to provide an example of what the assessment might look like. I did do a quick look through. I didn't see any obvious or consistent metrics that were significantly different from the reference group that would easily explain why the site is divergent between 2013 and 2019. You do notice that the abundance fluctuates from year to year. The highest values are seen in 2013 and a decline in the following years. This may be related to flow conditions within the Kettle River. It's hard to say with just looking at the community data. So we'd want to be bringing in water quality and hydrology to try and figure out what's going on. One thing I did note though, in 2019, we see a fairly large decline in the percentage of the EPT, like the sensitive taxa. So this might be a good idea to keep track of what's happening with the EPT over time. So here's the second site. So this is Kettle River at Grand Forks. It's located near the Raggett Road Bridge, again, for those that are familiar with the area. And the picture shows the upstream view from the site. So similar to the other site, there's data available between 2007 and 2019. And this site was also determined to be most similar to the reference group 2 sites. The results from this site are a little bit different from the upstream one. We see in 2007 and 2010 that the site is characterized as highly divergent as opposed to the upstream site, which was mildly divergent. So it's quite different from the reference community. We see conditions appear to improve in subsequent monitoring. The site is characterized as divergent. Again, I've got an ordination plot that just gives you an example of the output from 2019 and how the site actually compares in ordination space to the reference cluster in the middle. So I've highlighted some of the values in the table. Again, I'm just presenting this to show you what sort of assessment is possible. So I haven't gone in and done a really detailed analysis of the data. You can compare the results here with the average, the mean of reference group 2. You can also compare them with the upstream site because the habitat is similar at both sites. What I did notice when I looked at these, there's quite a lot of differences that we can see in 2007. So we see there's very low abundance. There's a higher proportion of the pollution tolerant taxa that Stephanie talked about. So the chironomids, the percentage of chironomids is quite a bit higher. We see a lower proportion of the EPTs. So it's quite different in 2007. In subsequent years, we can see the community shifting, and it's more similar to what we'd see at the reference sites. One thing I did notice, so Stephanie talked about going in and looking at the taxa that you expect to find versus the taxa that you observe. So we have at the top here a ratio that's calculated in cabin, like the observed to expected ratio. You can see throughout the monitoring that the value is low. So a value of 1 means all the taxa that you would expect are present. The value is quite low, so we know we are missing some taxa. And so, you know, if we had more time or we're doing a more detailed assessment, we'd probably go in and look at what taxa specifically we're missing from the site, and that would give us an idea of what's happening or what variables might actually be affecting the community and making it different. All right, so hopefully that presentation gave you a good understanding of biomonitoring and the cabin approach. It is one tool in the toolbox for watershed assessment. It's useful because it gives you a direct measure of the condition of the aquatic biota. Usually you'd use these data in combination with information on watershed stressors, the hydrology and water quality, possibly sediment quality, type of habitat present, and it provides a more complete picture of the aquatic ecosystem health. So there's some contact information here for Stephanie and myself, as long as provincial and federal web pages where there's more information on biomonitoring and cabin. So you can go there and find more documents and supporting information. Anyway, that's all I've got. So thank you very much for your time. Turn it over to you, Christina, if there's any questions.
Oh, I've got questions. Thank you very much. That was really interesting. And I think I found it interesting too, like in terms of the abundance, like the difference in abundance between the Kettle River site at Raggett versus Midway. So in that time, it's dropped down into the States and then come back up again. And yeah, it seems to be consistently significantly lower. Could that be a length of time, or is that standardized as well? So like when you're doing your cabin monitoring, could the abundance number be tied to how long you were actually monitoring, or that you'd be able to take that out of the question?
Yeah, I mean, the cabin, it's a timed kick. So you kick for 3 minutes.
Okay.
So depending on who's doing the kick, you might have someone that's a more vigorous kicker, might be able to cover more habitat. So I mean, typically total abundance is interesting because it gives an overall sense, but you know, it can be variable. Most of the cabin assessment, we're looking at the relative abundance of the different organisms, not the actual abundance. But I like to include it in an assessment because sometimes it really pops out. And in this instance, both those sites were sampled, I believe, by the same sampler. So the results should be comparable.
And do you try to do that with your sampling to make when you're sampling the same river to make sure that it's I guess if it's federally or provincially run, that it is the same sampler, or is that?
Yeah, in theory, it shouldn't really matter because there is
Okay.
the training. So the sampling should be fairly consistent regardless of who's collecting the data.
Yeah, that was one of the things that we looked at when we established the standardized protocols, is we had some experienced benthic experts doing this sampling. And then we took somebody from my office who was essentially a technician and said, you know, can you train this person who used to just do sediment work to actually collect bugs and be equivalent to these experts? And they were. So that was one of the ways where we said, okay, we've tested this. We know that if we train them properly, it doesn't matter who does the sample, as long as they've been trained to do it properly, we can get consistent results.
Oh, wow. We've got one question coming in from Graham over in the Q&A. How does a benthic invertebrate community typically respond to major flooding events or major increases in sediment input?
Two different questions, two different responses. But yes, flooding events for sure. You probably get like a big washout. So that's another important part of the standardized protocols is when we're doing benthic invertebrate sampling for cabin, we want to do it during the low flow season because we want the flows to be as stable as possible because we don't want that effect of the big flush. So we tend to do them kind of end of August. We might get into maybe Thanksgiving in some places, but then after that, the water levels are just, you know, it just rains too much. So it's number one, safety is a concern, access is a concern. And then of course it's the water levels are fluctuating quite a lot. So that's the first thing. We have seen, especially in recent years, some unpredictable water levels and flooding events during unusual times. For me, my experience particularly was in the north, so near Fort Nelson, where I'm just, you know, I'm new up there, sampling up there and going, what is the water level normally? Because I was there 3 years in a row and they were extremely different. And you could see in the abundance numbers specifically that we had to be very careful about making sure that the water level was stable for, you know, at least a week's time before going out to actually doing the sampling. So that matters. And then with respect to increases in sediment, that's the kind of thing that we kind of pick up over time. We wouldn't necessarily notice that very quickly. With the Fraser River, we know that there's a lot of sediment in the Fraser River. So one of the things that we have seen in our water quality results is, you know, we'll get a lot of sediment, metal exceedances associated with our Fraser River water quality sample, but it's okay because the bugs are used to that. So as soon as you start correcting for suspended sediments in the water column and that sort of stuff, you can kind of tell that those exceedances are related to sediment, but the bugs showed that everything was fine. So even though our water quality was showing a problem, the bugs were like, yeah, but this is my habitat and I'm totally used to this kind of sediment. So in that environment, it's totally fine. If there's a change in the sedimentation, we would probably see that and it would change in different ways. So one of the examples I mentioned before is, you know, some of the, like the pretty mayflies that have all those gills that have water just flowing over them, they'll get clogged by sediment. So you won't see them. And things like chironomids and worms burrow into the sediment. So you might see lots of those because I don't really care if there's a lot of sediment. So that's the kind of stuff you look at in the community when you're trying to pick up these things.
So one of the big ones that we've got here is based off a couple of recent fairly extensive flooding events. Yeah, just that change in sediment erosion and accumulation in different areas throughout the channels. What I'll quickly do is I'm going to put up a poll. So this was a poll that we wanted to look at.
Oh, we skipped one, didn't we?
Oh no. This is not a skipped poll.
Oh right, good.
This is just sort of looking at, you know, what are people seeing? Or yeah, how now that you've seen this presentation, and I liked it just because we did show at the very beginning a lot of people saying, I don't really know too much about the benthic populations or the aquatic invertebrates. So put that up, and then I've actually just got, and so I'd encourage everybody to give us a question. If you've got any questions, maybe some questions about what are things that you should be watching for in your streams. So I just sort of put out a couple of questions. I'm trying to see here. And I will just end this poll. And ending. Oh, sure. So there you go. We've definitely got quite a few more that are getting that familiarity. So of the benthic system. Are you seeing my screen in presentation form?
Not in presentation.
What I was starting to think. I'll quickly do that. So one of the things that you did say, you spoke a little bit about, is if people wanted to do it themselves, what would be something that somebody should call in? So let's say somebody, I don't know, was looking at the aquatic invertebrates in their stream and they started noticing a change. Is there somebody that they would call? Would they contact somebody to do a cabin monitoring? And how does a person sign up for this 2-day monitoring training?
Good questions. So first, I know DFO has a stream keeper program, which is a little bit more like, you know, I'm just curious to see what's in my backyard stream. So there's no real official assessment associated with it.
Okay.
So when we started this, we were talking to some of those stream keepers and said, okay, well, if you want to take this up a notch, so people would like pick, you know, they'd take their samples and they just look at the bugs swimming around in a tub and kind of say, oh yeah, I got some mayflies, I got some stoneflies. But this takes it up a notch. So it does require people to have some training, which I will mention in a minute where you can get it. And it requires you to preserve the bugs, which means you're going to sample them and you're going to kill them and put them in a jar and send them to a professional taxonomist to analyze them. So it does require some money and it does require some training. So that's why I say it's a bit of a step, an additional step to what the DFO streamkeeper work is.
Okay.
If you go to the, and I should have provided this in the presentation, apologies for not, but on the cabin website, which the link is there at the end, if you go to the training page, it will give you some information and it will direct you to the Canadian Rivers Institute page. So the Canadian Rivers Institute is actually administering the online modules for cabin training. And then there's a distributed network of mostly EC or Environment Canada trainers, plus some other external trainers that will do the 2-day hands-on training. And that will essentially give you access to the cabin database, the tools, all the information you need to be able to collect your information in a standardized way. I have to add a caveat to that is because we're still kind of in that COVID restriction situation. So there's not a lot of in-person training being delivered, definitely not by Environment Canada yet. But there are some other external trainers who have limited capacity. So I just wanted to make sure that everybody knew that caveat is still in existence right now.
I think that you're probably going to find that there's some interest in this area, especially with the information that Jolene was presenting. I just think it really sort of stood out. One of the things that I hear a lot about is an increase in river snot. And I'm going to screw up on the actual name for it.
Just call it Didymo. We can
Thank
stop
you.
there.
Didymo. There we go. How does that impact the benthic? So that's something that we're seeing a lot in the Boundary Creek, very much in the Kettle. And so in the sites that you're speaking about, the Midway, I know it's Raggett. We've got a lot coming in there. And one of the things that we've also been hearing a lot about lately is drought. And so being able to try to understand how that would all.
Didymo is interesting. We spent a lot of time with looking at that with other EC colleagues as well as Parks Canada, because we were concerned that in our waders traveling all over the province that we were actually one of the problems spreading this stuff. But it's an interesting, if anybody does any kind of internet searches for Max Bothwell, he's an Environment Canada scientist who's done kind of a lifetime of research in Didymo.
Okay.
It was a long time where we thought, okay, it's the fishermen spreading this stuff on their waders, their felt bottom boots. And what Max had found later was this stuff actually has always been there. There's just a change in the ratio of nitrogen to phosphorus, which happens for whatever reason that causes the bloom. And so when the bloom actually happens, it covers, it mats the rocks. It's disgusting if people have actually seen it. It looks like wet, soggy toilet paper covering all of your substrate. And that's a problem for the bugs because the stoneflies and the mayflies that like to cling to the rocks, like there's nothing to cling to. They can't crawl on the rocks. They get suffocated by this rock snot. So that is a problem. You asked another question. One was about rock snot and one was about drought.
Oh, right. Yeah.
Drought is a problem too, because the organisms need flow, like especially those black flies with their fans in the air and the caddisflies that stick their heads out to try and catch stuff as it's flowing by. If they don't have adequate oxygen or adequate flow, they're going to disappear. So disappear meaning they will just release from where they are and drift to a more suitable habitat, or they will die. So those are the repercussions of drought, and you will definitely see effects from that.
Got another question for Joe, but I'm first going to speak to Alan's question here in the Q&A box. How many samples are required in order for the data to be considered statistically trending?
Aha, a science question or stats question. Cabin does status. Cabin doesn't do trends. Having said that, so our water quality folks do a lot of trends. So they want a minimum of 10 years data before they can do a trend analysis. Kevin hasn't really been doing a trend analysis. However, what we have done, and Jolene mentioned this, where we have sites that we will go back to over a period of time, we actually try to see what that temporal variation looks like. We know that communities will respond to a wet year or a dry year or a hot year or a cool year, and we'll kind of see that community moving. So if that cloud of reference sites is the baseline or the benchmark, we might see a site kind of circling like this, but it eventually comes back to its point. So we do see a bit of variation, but we don't really track trends. So there's no statistical trend associated with the cabin data. Hopefully that answers Alan's question.
So Alan, I'll let you know, let us know if that answered the question. So who's looking at this information? So when you're doing these sites over, so the Kettle River, or yes, you had a number of reference sites through the boundary. Who's looking at it?
Lots of people. So primarily Jolene and I, and the bigger governments like Parks Canada and stuff, we like to look at the reference data and make sure those models are maintained and kind of track that temporal change just to see if the model's kind of shifting in a particular direction. Mm-hmm. But in terms of your test sites or the sites that you want to assess, that could be anybody. Okay. So I showed you that industry is quite a big user of the CAP.
That's right.
So they go to a lot of sites that they've been hired by, consultants have been hired by an industry to do impact assessment or environmental effects monitoring, whatever, and they will use the cabin reference data to assess their site. So they're looking at their own site for their own interest, their own purpose, their own study design. But sometimes they also are, starting up before a new mine will start up. So they'll collect some really good reference data for us in that pre-development condition that they allow Jolene and I to use in the reference model. So it's again, that's that network of networks. Everybody looks at their test sites for their own interests. We try to maintain, Jolene and I, the BC models, and we will talk to other network participants to help participate in that.
Yeah, I was going to just say, yeah, most of the work that we do is primarily focused on collecting reference data and building the models. Another part of my job, I'm also helping develop water quality objectives. So this is like site-specific guidelines. So we are doing that for the Similkameen River. And so, having the consistent data has been great, because I can use cabin data that Stephanie has collected at the federal provincial sites on the Similkameen. There's data collected by Copper Mountain Mine, there's provincial data, and because it's all collected in the same way, I'm able to, once I get permission from the study leads, I can go in and actually analyze all the data using the Okanagan model and get a picture of what's happening in the entire watershed, and I don't have to go out and collect all the data myself.
Wow. And so is that data publicly available, or that would be, so the data coming in from industry? So
the data in cabin, and Stephanie can speak to this, there's been a shift. So the data has shifted to become open. Okay. So the data, it used to be like all the government data is open and available for anyone who wants it. In order to actually go into the industry study, I did have to get permission from Copper Mountain Mine, so I could actually analyze it using the Okanagan model in the cabin web tools.
Oh, wow.
Yeah.
So then if somebody was interested, so one of the things that it's spoken about around here a bit is the impacts of, say, leaky septic systems, or, so I guess there's sort of that nutrient inclusion into the water. or it could be irrigation, over fertilization of lands or lawns. Is that something that we'd be able to pick up through this type of aquatic invertebrate monitoring?
Yeah, the bugs are quite sensitive to nutrients, especially. So there's a thing about nutrients is you might see like a nutrient enrichment effect where you're like Jolene showed you, what you expect to see is in the range of like 4,000 bugs or something at the kettle. A nutrient enrichment effect might mean that you found 10,000 bugs at your kettle, where you're like, whoa, and that's going to fall into that red zone in the graph. And you might see all of the bugs you expect to see, but it's going to be in way more proportion than you expected. So, and that's usually our nutrient signal. So that big pulse usually happens first before things start to die. So they're pretty sensitive to nutrients that way.
And it's interesting as well too, because nutrients are taken up very quickly. So you may be able to detect them in the water for a very short area, but they're taken up, by the algae, like the lower trophic levels. So you can collect a water sample downstream and you may not pick up the phosphorus from that leaky septic system, but you're going to see, you can measure, the algae. As an example, you can look at the chlorophyll A or the productivity in the algae. You can look at the benthic invertebrates and you can see the change, whereas you may not be able to actually pick it up in a water sample.
Oh, wow. So then I guess this sort of comes in a bit to the drought question. If you've got a stream then that goes dry, let's say it's a losing stream, it's gone totally dry. Well, I guess if they can go flying, I'm somewhat answering my own question. How well is the aquatic invertebrates impacted by a totally dry stream? Say then the next year you go to sample it, are you going to notice it that stream went dry, or is that, there's enough resiliency within the organisms that they're able to manage that? Again, I'm back to that drought question.
They're quite resilient. Like they will, like one of the things that somebody always asks me is you kick the stream and the substrate, you're disturbing the substrate for 3 minutes. So you're scraping, you can see where you've kicked in the stream because all the rocks are turned over in the algae, and they're like, ooh, you've really disturbed this. But it's not long before the bugs have moved back in and recolonized. So kind of depends on the colonization. So one of the streams that Jolene and I were trying to incorporate as part of our routine annual sampling was it West Goat or Little Goat Creek?
Big Goat or Little Goat?
Something. It's a tributary to the kettle, I think. And every time we went there, it was dry. And we're like, this is not working for us. But if there was a site that you were interested in sampling that didn't always go dry like that one, it depends on how long it's dry, because there are bugs that live in that kind of, not really the groundwater layer, but the hyporheic layer where they're small and they'll live down there and they're fine down there as long as it's wet. But there's others like the mayflies and the stoneflies where they can't live that way. So they need to go find other water. And then when it fills back up, they'll eventually just kind of recolonize. So it depends on when you're sampling. If you're sampling it right after it's just been wetted for the first time in a month, you might not find very much. If it's only, dry for the couple weeks in the summer and then it's, wet again in September, you probably find lots of things there in a couple weeks time.
This has been great. And I, of course, I want to look into a little bit more on the changes between the two sites for the kettle. I think that's been pretty interesting, and also sort of where some of your other sample sites are. Does anybody else in the attendees have any questions that they would like to ask? I would like to know if Stephanie has any caddisfly jewelry.
I do.
I knew it.
I do. And if anybody's interested, the woman that I buy it from, I guess this makes me officially nerdy, but it's called Wildscapes. I think she still operates out of the US, Wildscape. But I used to go to a conference. It's a bug conference, I'll admit it. And she would always have her jewelry there. So yeah, I would buy jewelry from this woman at the bug conference.
when we were looking at trying to put up the poll, and I didn't have caddisfly on it, and somebody's like, "You have to. They made a story
That's,
on
what,
caddis."
that's my favorite. Caddisfly is
You
my favorite.
have to add caddisfly.
There, Ellen found it. Wildscape.
Thank you, Alan. So this is what it looks like. Well, I just want to say thank you very much. If there's no other questions, I know that this is certainly something that I need to spend a lot more time learning in. The Dynamo is a big one for this region and better understanding the impacts of that. I know that's something that is spoken a lot about from some of our longtime residents in the area, just seeing a huge increase in this region over the last, say, probably 15, 20 years. I think there's a lot of interest trying to better understand where are the concerns and what can we do. Are you noticing anything in terms of the riparian protection? Does that seem to have an impact on it? And do you look at, does a cabin look at that to how much protection is required in order to have those types of impacts? Is any of that tied in with some of the riparian monitoring? Do you know?
It's not specifically, but it is one of the variables we measure on our field sheets, where we say, what does the streamside vegetation look like, and the kind of stuff. And it matters. It matters a lot. Because we have been to streams where there's been no riparian, and we've been to streams where there's been a lot of logging, but as decent sized riparian, and you will get a decent community as long as it's got a protected riparian.
How long, if once impact has happened, and then there's some efforts to do that protection, how long, I don't know, is that even a question? Can you even ask that question? It may be so site specific. How long to get the reestablishment of the aquatic invertebrates? So yeah, that's sort of why I struggled when all of a sudden I started realizing my question is so completely site specific.
It'll be site specific. It'll be issue specific. It'll be specific to 1,000 things. They do recolonize quickly, but do they recolonize to what it should be? Because it will go through stages, right? Those opportunistic organisms, and then it'll eventually stabilize. So that's a hard one to. When
you're asking a question and you know it, you hear it in your back of your head. So Martin is saving me here with a question in the Q&A box. In using these data to complement watershed assessment and to better understand watershed health, it seems important to know how responsive the benthic organisms are to changing in water quantity and quality. For example, how quickly do they degrade, go up or down, and then return to normal levels? Somewhat similar to the question. I was asking, how variable are they up and down a stream? And can you speak practically how this data can actually be used to interpret watershed health, particularly in relation to land use? Well, Mark,
that's a big question.
You and I are on the same program. So I don't know if can you see the question on your side? Okay, good.
Yeah, thank God, because I'd have to ask you to.
That's why I was asking.
So it does respond to water quantity, and it is one of those things where, because cabin, this is something I didn't mention, cabin goes out to sample once a year. So our water quality, I mentioned we might go out bi-weekly or we might go out monthly. For cabin, we go out during the low flow season once a year, because bugs are seasonal. So I didn't really get into winter stoneflies versus spring stoneflies, but bugs emerge at different times of the year. So it's really important that you always sample at the same time of year to make sure you're comparing apples to apples. So we, for cabin, usually go out kind of late August to Thanksgiving. So that's usually our window. It might shift a little bit as you go north, but generally that's what we're talking about. So we know we're comparing the same one. And in order to see how that quantity, how quickly they recolonize, you probably need a special study to just kind of track that with water quantity. We haven't really done that, but we've done lots of different sites where okay, we know that nothing else has changed here except the water level is really, really low this year. We do have an example of that on our cabin website, where we looked at the Quinsam River when we first started to build cabin into our long-term monitoring. The Quinsam River on the island was the reason we were monitoring there for water quality was because there was a coal mine upstream, and they were concerned about changes to water quality, and they hadn't really detected anything. But when I went there and collected cabin, the community was way, way different than what we expected. So there was a lot of concern about what was going on, what was the water quality missing. And so we tracked it for a little bit of time, and also did a bit of a longitudinal study throughout the Quinsam. And what we found, and that was probably the first time we actually found that there was lots of things going on that wasn't being picked up by the water quality. Like there was a big flood event the year before we sampled. And so I think what we were seeing was the effect of that flood. And then it stabilized, and then it got better over time. The other thing I think we picked up is they had a super productive fish year that year. And so I think there were a lot of nutrients in the system. And so there was all these really interesting things that kind of set the ecosystem kind of off kilter, but the water quality samples weren't picking that up. So an example of that, and that was quite a few years ago, an example of that on the website, but it wasn't really designed the way Martin is describing here. You kind of would have to do that to kind of see how quickly does this happen. The next question was how these data can be used to interpret. There was something else I wanted to address in relation to land use, degrade. There was something else that I lost my thought, but we are using this in combination with our water quality data to kind of describe in a more complete picture. Like water quality says everything is good, bugs are actually looking pretty good, so things are okay. We're tracking the site every 3 years so we can see if if anything changes. We'll track it more often if we know that something different is happening in that watershed. Like when the Olympics are happening in Whistler, we said, okay, let's start keeping an eye on this more frequently. We're not doing this every 3 years, we'll do this every year to see how the development affects this. And so it's used in combination. It's kind of weight of evidence. So that's kind of how it's used to interpret if the ecosystem is in its status, right? So it's in a good condition. And then when it's in bad condition, we have to dive deeper. We have to do more investigation.
So it almost seems a bit like it's just because, in doing it, say once a year, it sort of seems like it helps to correlate what you are guessing is happening, or maybe helps you to sort of, so if the water quality data is off, or we think there's something strange going on, is there actually something strange going on?
Yeah.
And being able to speak to that. I'm just thinking, like, if you're doing it just once a year, then it would be more of a one shot.
Well, it's once a year, and that's the community you're sampling. You could do it a couple of times, but that was the other thing I was going to mention, the variability within a site. So when I talked, I showed you the picture, and probably people didn't pick up on it because I didn't talk about it, was we zigzag through the site when we collect our sample, because we know that if you just did a sample right here in the stream and a sample right here in the stream, those could be different. We know that there's going to be variables. So the way the cabin is protocol standardizes, we zigzag to capture all that stuff in a single sample. So the whole point is to try to incorporate all of that variability into our single sample. And if you wanted to do that this week, and again next week, and again the week after that, and we've done that, it's going to be pretty stable. It's going to look pretty similar, as long as there's been no changes. Okay.
So I have one last question. What's the discharge mechanisms you use in terms of how do you monitor discharge? So one of the comments you've made is coming to a couple of these sites, suddenly saying, the water's way lower than what I was expecting. I'm used to seeing it higher, or it's just that big variation of water level. I'm thinking that the benthic, the aquatic invertebrates would also be very much tied, as you were saying, to what that water level is. What do you guys use
we do
discharge?
I need to find my outlet. So if I cut off, Jolene, you can fill in, right?
Yeah.
Realize my battery's dying. But we do measure a lot of the variables needed to get a rough estimate of discharge. We measure widths and depths and velocities at multiple points, so you could do a rough calculation for discharge. Our long-term water monitoring stations are usually co-located with water survey sites. So if we need that detailed, we can get them at those sites. Obviously, all of the reference sites don't have that, but we do have that information, so it could be calculated.
Nice.
Yeah, I'd say if flow was a concern in a stream that you were sampling, you'd probably want to set up a station where you're measuring water quantity.
Okay. Yeah. Thank you. Thank you very much from the Boundary region and from the community here. It's been super informative. I really appreciate you showing us the levels and the information on the Kettle River. And I know that I'm certainly going to go to your website and take a look at what else is out there. And hoping to be able to learn a little bit more about how to do this in this region. I think it's a really great way of looking at what's happening and being able to see some of that as that concern or not a concern, or just that variability. And shucks, I think I just need to learn a bit more about my aquatic invertebrates. So this would be the end of the webinar here. I really appreciate Jolene and Stephanie for joining us on this and sharing this information. They did provide their contact information. If you've got any questions, feel free to reach out to them. I know that they were great at responding back to me on taking on this task of webinar presenting. You will receive a survey as you shut down from the webinar. It's the same survey you're going to receive tomorrow, just as a follow-up. But it's just giving us a little bit of information about how you heard about it and about what works, and some ideas on what you got out of it. And I know that we really appreciate hearing from the Boundary folks as well. So thank you very much. Have a great afternoon, and we look forward to seeing you in the fall.
Hopefully in the river.
Yes, I would actually be interested in finding out when you guys were coming down this way. Oh, so
I'd be interested to find out when I'm allowed to go down that way too.
I know. Well, this is actually one of the comments I was going to say to people. For those non-boundary people, come visit us when the COVID restrictions are stopped, when we're able to do it. So, okay. Have a good afternoon. Okay, thank you.
Okay, thanks,
Bye,
everyone.
everybody. Thanks.
Bye.